Okay, good afternoon, everyone. We will start the class, and we were discussing about site-specific recombination. In that, I think I may have missed it. We were discussing about recombination that happens between specific sites. It's like fragments of DNA mixing up, right? That is the recombination, and there is a change in the information. So what I want you to also remember is it is called as conservative site-specific recombination, right? Why is it called? This is fine. Site-specific recombination is fine. We understand that. That means it happens between specific sites. But there is something about conservative. Why is it called conservative site-specific recombination? And that is because it says it is conservative because we should count the number of bonds broken. Number of bonds broken is equal to number of bonds formed. And when we are talking about bonds here, we are talking about phosphate bonds, right? So when we are also learning about this, we learned about two types of recombinases. In that, we learned about serine recombinases and tyrosine recombinases. It is based on what kind of residue is involved in the active site that makes the nucleophilic attack. Right. The one more feature of these recombinases, in most of the cases, is that they form these protein DNA. They form protein DNA conjugates like this, intermediate structures. I want you to tell me if everything is fine. You can visualize the board and you're able to hear clearly, right?
Yes, sir.
Yes, sir.
Oh, thanks. So what we are doing now is that we will just go review the serine and tyrosine recombinases, and then we'll see transposons. That's something very interesting also. So the difference between we will discuss between the differences between serine recombinases and tyrosine recombinases. In serine recombinases, in all the cases, I hope you remember these structures. These are recombinase recognition sites, the blue ones, and here the orange ones. And the middle one is called the crossover region. So these recombinase recognition sites are repeats. They have same sequence. When two are aligned, when two sites are aligned like this, and recombinase will bind in, say, two subunits here and two subunits here, and then they make a double strand break or single strand break based on whether it is a serine recombinase or a tyrosine recombinase. Serine recombinase makes a double strand breaks, whereas tyrosine recombinase makes single strand breaks. In serine recombinase, they just exchange the whole double stranded. The orange one goes up and the gray one comes down, as is shown here. And then these broken pieces are religated, and then you have products of recombined products. In tyrosine recombinase, it happens one step at a time. There'll be single strand break, then the formation of a holiday junction, and then the second strand break, and then the whole exchange. Okay, so it is conservative site-specific recombination because you have the number of bonds broken is equal to number of bonds formed. You have 1, 2, 3, 4 bonds are broken, and eventually when the product is formed, 4 bonds are formed. The same is the case here. You have 2 bonds broken, then later 2 more bonds broken, 4 bonds are broken, and here you have 1 bond formation, here you have another bond formation, and in the completed product you have 2 more bonds formed. So it is conservative, and that is with regard to the number of bonds broken. Okay, we also had an interesting example about heme recombinase. We discussed in detail about this Higgs L, and every recombinase comes up, recognizes specific sites, And here, hin recombinase is recognized by HIXL and HIXR. So the organization is something like this: FLJB and FLJA. A is the repressor of H1 flagellin. FLJB is secretes or actually encodes H2 flagellin. Those are the molecules that are the proteins that are required to synthesize flagellum. Okay. So whether a salmonella has flagella that is made of H1 or H2 will depend on the orientation of this invertible segment. Okay, recombination can occur between HIXL and HIXR, and if they are present in opposite orientation, right? They're not in, both are not in the same. And if recombination takes place between them, you will have the orientation of this promoter either upstream of FLJB and FLJA or away from them. If there is no promoter, these genes will not be expressed. If they are not expressed, we will just see the consequences of what happens. So in this case, say promoter is here, so FLJB is expressed, therefore you will get H2 flagellin. And there is also something else. There is a FLJA repressor. So H1 is not expressed, right? Because this will go and repress the gene of H1 flagellin. If recombination occurred between HIXL and HIXR, then the promoter is away in this way, and there is no expression of FLJB or FLJA. And then because there is no repressor, somewhere in the genome there is H1 flagellin, and that will be expressed. And H2 here is not expressed. So the flagellin in this state will be, the flagella is made up of H1 flagellin, and in this case, it will be made of H2 flagellin. So using recombinase, Salmonella can alter between having flagella made of H1 or flagellum made of H2 flagellum, right? You will find many things that happen in the throughout the biology. You can take any example, actually. You will find so many of these things, and then you have a phenomenon coming. Here, the phenomenon is the ability to change from H1 flagellum to H2 flagellum, right? Some of them are very well understood, some are still being studied. So we will look into something called transposons. You heard about them, and it is a question of philosophy. It has yielded in Nobel Prize to Barbara McClintock. You should also try to know about the lady. She's a fantastic scientist. And when she proposed a scenario like, I mean, it will look absurd even today to me, that if somebody were to come and tell us that there is this DNA and there is a gene here, and that gene is moving from one location to another location. If somebody said that, it would be absurd. Even if after these many days, and we suppose didn't have the idea of transposons, and somebody comes up and gives this, it feels like crazy. But Barbara McClintock was not encouraged because this idea seemed too wild to be true. And that is the historical perspective, historical significance of it. And she's a woman, and she came up with a very weird idea. And science is typically dominated by, as in many fields, dominated by men. And then she was not heard well. And eventually she kept working on it. She didn't give up on her idea. And eventually it earned her a Nobel Prize. That's a fantastic achievement by her. And how did one conceptualize this? And the ability to move of a physical entity to move from one location in the genome to some other location. How can that happen? The mechanistic problems are there. And some second thing is, why? Why should they move? What is the use of such movement? Is there any use? Are they part of the genome or are they not part of the genome? As I was describing sometime about the definition or tentative definition of what is genetic information, right? I said there is there are two things, if you can recollect. Genetic information has to have two important things. One is it should code for something, a RNA, a protein, or some structural thing, if required. The second thing is it should be associated with a replicator, right? It should be a part associated with a replicator because origin of replication is important for replication of everything else, right? If you had a gene in the cell like this, and it may give rise to a protein for a while, but when the cell divides, and it will get degraded. First thing is it will get degraded. Second thing is it is not associated. If it is not associated with the replicator, then it will not be inherited by the daughter cells. When I say replicator, all bacterial chromosomes have origin of replication. All eukaryotes have something similar, that is autonomous replicating system. You can have plasmids, they have their own origin of replication. You can also have viruses, and they have their own ways of replication, or viroids too. Everything has a way of replication. So the problem is with genetic information, right? And here transposons are these are called as transposons. These genes can move from one side to the other side, and there are two broad mechanisms. And why I brought up the genetic information is, say for example, this is a chromosome and this is the origin of replication, and we have a gene here, right? And if this chromosome replicates, it will form two new chromosomes, and you will have two genes of these formed as well, because they are associated with this replicate. But what if the replication or the propagation of a gene is not proportional to the origin of replication or to the associated replicator? This is the origin of replication, and here is a gene, right? The number of origins will typically be equal to the number of genes, isn't it? More number of replication, you will find everywhere these two. Now let's see something little bit different. You have a transposon here. Say, I will just go along with this. This is a transposon, right? It has a definite structure, and it encodes for certain genes and elements. We will discuss that in a later slide. Just assume this is an introduction to it. One of it is called as cut and paste method. Yes, we all know, we all are very proficient in using Control C, Control V, Control X, and Control V, right? Cut and paste, or we have another one that is called as copy and paste. So transposons can move from one part of the genome to another part by either of these mechanisms. Some of them go by cut and paste, the others go by copy and paste. Okay, we will see those, and then we will come to some more discussion. At the end, we all should with living organisms, it has to do with two things. One is survival, okay, and the other one is evolution. You should have this thought Always when we are finding an answer to why in biology, it should reflect in either or both of these things: survival of the organism and evolution of the organism. Typically the answers will be there, but it's not certain, it's not limited to them. So if you look at how many transposons are there and how widely they are distributed, you can all the green ones are the transposons, the blue ones are their genes. Okay, here is E. coli. It has very few of them. So here they are written as IS. It is for insertion sequences, right? Typically in bacteria, you call them some of the simplest genetic elements that move around that are transposons are called as insertion sequences. With yeast, Saccharomyces cerevisiae, you have a larger number and they're actually larger in size as well. In Drosophila, you have many more, and with humans we have smaller, and they are numerous in number. And if you, sorry, I'm pointing the wrong one. Sorry, the green ones are the transposons, right? Yes. So you have many more in human genome. And look at maize. You find them like they are. It's all full of transposons. I think Barbara McClintock worked on maize, and she was working on the color of kernels. If possible, some other time we will discuss, but it might be too much at this point to go to research level. What we will discuss is the mechanisms of how things happen. And I also have a question. Are you studying immunology now, or is it going to be next semester? Anybody, please answer.
We are not studying it mostly.
You're studying immunology now.
No, sir, no.
Oh, is it in the next semester?
We're not
Probably.
sure, sir. We heard some.
Okay, okay. It will be in the next semester then. Okay, I'll see how far we can get then. Okay. Yes. So there are three classes of transposons. One of it is called as DNA transposons. Second one is virus-like retrotransposons. Try to remember retrotransposons. And the third one is poly-A retrotransposons. Whenever you see something like retro, that means you should try to understand that an RNA intermediate is involved. Okay, try to remember that. And that is why these are called DNA transposons in which RNA intermediate is not there. I will tell you what is meant by RNA intermediate here in this case. The first thing what we will understand is the architecture of the DNA transposons. So all transposons have these things. There are types. First thing is they should have the gene for transposon. Okay, there is assume that this is the gene for the transposase enzyme. What is the enzyme is the enzyme that mediates the transposition is called as transposase. Transposase. And there are something else, and they are essential. What is meant by essential? If a DNA molecule has to move from one place to another place, they should have these border regions. Okay, and these are called as terminal inverted repeats here, or you can just say it as inverted repeats. Okay, and sometimes you also call them as left and R. So this is coding for transposase enzyme. So these are the basic elements that are required. There may be other genes or they may not be there. Okay, as we will see in the examples further, we will learn more. And there are also certain other sites which are not part of the genome, but which are not part of the transposon, but they are part of the genome actually. Okay, these are called as target site duplications. We call them as TSD. I will tell you how they will form also, TSD. So if I clearly have to write it, from here till here, it is transposon. Okay, what are the basic essential elements? You should have the inverted repeats. Those are the essential ones. There is another essential requirement, that is the gene for transposase. Say, for example, this gene is not present. Transposase is not present, but it is present somewhere else in the genome. Assume that transposase gene is present somewhere else. If it is expressed, transposase enzyme binds to these inverted repeats like this and cuts the DNA and puts it into a different target site. Okay, that is the function of transposase. We will see in the next slides, but just remember that, right, for this purpose. This slide is to bring you some terminology so that you will remember what you meant. Autonomous and non-autonomous. If a transposon has all the elements required to move from one place to another place, then it is called as autonomous. What is required to move? You need the inverted repeats or terminal inverted repeats, right, on either side. That is essential. And second thing to move is the enzyme, the gene for transposase. It doesn't require anything else. You need an enzyme, a gene for an enzyme that should produce the transposase, and inverted repeats that can be recognized by that transposase. Okay. So if the transposon has everything, then you call it as autonomous because it has its own every requirement for transposition to occur. Say, for example, because of some error or some mutation, this transposase gene is mutated and becomes non-functional. Is it still a transposon? Yes, it is a transposon because the repeats are there, and using those repeats, they can move. But they can move if only the transposase gene is being produced from somewhere else. From somewhere, a protein transposase enzyme is produced, and it is not within the boundaries of the inverted repeats here, right? And then those are called as non-autonomous. Cis and trans we will discuss, and say there is a transposon, another transposon in the genome, and it is very much similar. IR inverted repeats are there, and it has the transposase gene also, and it can produce transposase, and that transposase can come and work on this transposon. Then this transposon is called as helper transposon because it is providing the enzyme transposase which can act on this one, right? There is something else I want you to also understand is, say, for example, some mutation happened, and you can also assume that this, say, for example, this is an autonomous transposon, its transposase gene is fine. And think of it like there is a mutation, and that mutation has happened on one of the terminal repeats is lost. What will happen if the terminal repeat is lost? Then, say, from here, transposase is produced, right? Transposase is expressed, produced. It can bind here, and there is nothing to bind here. And in that case, this transposon can no longer move from its location to any other location. Those transposons are called as grounded transposons. They are almost in the process of, in a way, dying out, right? If a mutation occurred in either of the inverted repeats, then that transposon cannot move any further. Okay, I hope you understood at least what is meant by autonomous. If a transposon has all the components required for transposition, then you call it as autonomous. If a transposon is functional, but it does not have the gene for transposition, then you call it as non-autonomous because it is depending on some other transposon, that is the helper transposon. The next one is grounding or grounded. It's like a flight is there, it can fly and go around and so on. But assume that it's one of its wing is broken. When it is on the ground, wing is broken. It can never fly again. And then you will say it is grounded, right? So the same way, these are also referred to as grounded because one of the terminal repeats, one or both of them are mutated. Okay. Yes, let's go and see some examples on cut and DNA transposons. First thing is as I said, the requirement for it is to have inverted terminal repeats, and the transposase enzyme recognizes these repeats, and then it will bend the DNA and bind something like this, and then makes double strand breaks like this. We're learning about cut and paste mechanism. Cut means it will cut off from that side and go to a new place. So when it is cut off, and these ends are going to go and attack a different site here on a different target DNA. What also happens is these two are the original sequences of the genome, right? The host site. These two get directly bound together, something like a double strand non-homologous end joining, and then you get the insertion. The DNA was here within this gray DNA indicated here, and into a new, that is blue, target site. And then DNA repair synthesis takes place and repair happens. And then you have the formation of target site duplication, like here and here. I'm sure you may have difficulty, but we will try to draw it out so that you will have a better understanding of what's happening. So let's try to write it. First thing is I will take a sequence. I assume that I'm talking about this step. Okay, I will write a target sequence: GACTAGTAC. And so the complementary strand is CTGATCATG. Okay. Now transposase is coming with this DNA molecule. Okay. So what it does is it will break the DNA somewhere here. Assume that it makes a break in this location like this. So what will you get? If I can make 1 second. Let's see. And let me see if I can pull these apart. Sorry, let me try. I can make it. Okay, I think I was successful enough. Okay, now I'm separating the DNA like this. I should have done it a little more, but assume that this DNA is far apart. This is for understanding purpose. Now I'm going to bring the transposase has come and put the DNA here. So the DNA is, the transposon is brought in and it is put here like this. So assume that this is the one of the target inverted repeats, okay? And this is the other inverted repeat, and you have the transposase gene here and so on. Okay, now what happens is there are gaps here. You can see that the gaps are in this place and so on. So the repair mechanisms can fill this gap like this and then seal it up. Okay. Now I want you to find out something, the difference between, this is the actual donor, the target site. This is the target site. If you look at closely this sequence, the sequence that I'm drawing, GAT, or I have to read on the top strand, CTA, GT, okay, this sequence. How many times is it in the bottom after transposition has occurred? CTA, GT, CTA, GT. Again, you find it here, CTA, GT, right? They are in this one. That is the duplication that has occurred, right? That is the duplication has occurred at the target site. And so you call that as target site duplication. Or TST. Say in one of the assignments that will come for the compulsory assignments, if I give you a DNA and it has a repeat something like this, GACTA, GACTA, GCT. Say if I give a sequence like this, you should be able to look at it carefully and analyze the sequence and be able to provide a logical explanation of what is it, right? We should be able to look for something here. So I tried to put a ACT-AG, and again I put ACT-AG. If you had this, what does that mean? Now, why I'm asking this question is, say, for example, if the transposon should move out from this location to something else, okay? If this transposon, by using cut-paste method, if it has moved from here to somewhere else, what would happen to these two sites? What would happen? It will come, so GACT-AGT, and then you have again this part, right? It will be CTAGTAC. That is what will happen after the transposon moves out from this location to a different location. GATC, AG, ATC, ATG. You look at it carefully, you will find that CTA, GT and CTA, GT, right? You find a signature that there was a transposon here, but now it has moved out. Anywhere transposon jumps in and moves out to a different place, you will end up forming these target site duplications. Okay, so when a sequence is given, I want you to look at it and try to incorporate all the knowledge that you have gained from different parts of this course and be able to analyze the DNA or the genetic information in that way. Okay, I hope you understood this. Maybe I'll repeat it in the next class just for a review. But have a look at the textbook, and it is a very interesting chapter in many ways for understanding for the wonder of transposon itself. How can this happen and why does it happen? Well, so the molecular mechanisms, it is very much similar to many things that we see. We have seen this. This is the 3' end of the transposon. Sorry. Okay. So this is the 3' end of the transposon. That is this one. Okay. It is coming with this 3' OH, and it is making a nucleophilic attack on the phosphate. So this DNA will be broken in, and the transposon becomes covalently attached to this. Okay, and then we have some things understood and some things that we don't understand. This is first strand break. How does the second strand break happen? And there are 2 or 3 mechanisms because of based on which DNA transposons are further classified. So in one case, transposase cleaves DNA like this, and there are 3 different examples, right? So see what is the difference between them. You don't have to by heart it, but it is good to appreciate that it is not just one mechanism, and there are many ways of how transposition can be achieved. So first strand break happens this way, and then what happens to the next strand is the question here. There is a transposon called TN7 in bacteria, which also encodes on the protein called TNSA. TNSA breaks the second strand, and then you have the whole DNA. Then this whole transposon is taken to the target site and incorporated. In another transposon called TN10 and also TN5, transposon 10 and transposon 5, you see this OH and you try to compare with what happens in TN7. The first strand breaks are made, and the OH here makes a nucleophilic attack on the second strand, okay, forming a kind of hairpin loop here, like this. Okay, there is no extra protein involved. It is transesterification. It was here broken and a new one is made. And that hairpin is eventually resolved, and then the transposon moves from the donor site to a target site and gets incorporated. In Hermes, there is a different mechanism. It is opposite to what happens opposite to TN10 and TN5. So the break happens here, and the OH of the host site makes an attack on the second strand. So you're having this transposon is free and will move on to the target site, and here you have the hairpins on the donor site. The donor site will eventually be resolved and they will be joined together. Okay, so what you should understand is this is also called as the non-transferred strand, which is the second strand that is being degraded. One second, sorry. Yes, so cleavage of the non-transferred strand, that is the second one. Okay, so first cleavage, and then in some cases you have the specialized proteins to make the second strand break. In another, the OH will make a nucleophilic attack and forms a hairpin. Hairpin is resolved, and then you have transposon that can move on to a new target site. Similarly, we have also the OH of the donor site could make a hairpin like this, and the transposon is free to move from that location to a different location. So there is also something called as replicative mechanism of replicative transposon. In this sense, here it will move in a little bit of a little different, or it is associated with replication. So here is a transposon on a DNA molecule, which could bind to, it has made only single strand breaks here and here. And those single strand breaks are making a target site or attacking the target site. When they happen to form something like that, they attack, an intermediate like this will form, where the attack, it's like the transposon has already integrated with the target site, but it has not shed the donor site yet. Right? This is the donor DNA molecule. This is the target. So you find a cointegrate like structure that has formed here. And then replication fork is formed. Here is the replication fork. You see that, right? Assume that this one side of it, you can see that there is this can be something like a leading strand, and you will find basically you have all the requirements to form a replication fork here and here. And the DNA will keep opening up on either side, a new DNA molecule will be synthesized. As a result, what happens is that you have replication fork and you have two strands synthesized. What are those synthesized? It's actually the transposon that gets synthesized. And eventually, in between these two DNA molecules, we had only one transposon. This is the only transposon that is present between these two DNA molecules. You have one donor DNA molecule, one target DNA molecule, and one transposon. Look at the eventually. The donor DNA molecule is here, one, and you also have the target DNA molecule, that is one. What has increased in number is the transposon. There was only one transposon, but now we have two different transposons, right? It has replicated, and it is not in line with the replication of the the target DNA molecule itself. So that is called as replicative transposon, right? It involves replication, and through replication, it increases its own number, right? Others have remained as such, but the transposon has increased its number. So the second one is called as retroviral integration. I will just describe the structure first, and then probably I will stop there. I will just describe these two structures, and then we will discuss the rest of it in the next class. So you have the DNA transposons we have already seen. They have inverted repeats. And you have a transposon gene, and there are target site duplication. One thing common you will find in all these transposon is the target site duplication. Okay, you see these blue arrows, that is representative of target site duplication. So the second category of transposons is called as virus-like retrotransposons or retroviruses. So they are similar to that of, or many viruses also form similar, they involve similar ways of replication. So there is a long LTR, long terminal repeat. There is a gene for integrase and a gene for reverse transcriptase. RT is for reverse transcriptase. I hope you remember what is reverse transcriptase. Reverse transcriptase is a gene, is an enzyme. It's a polymerase, like we have seen, for example, when we were discussing about telomerases. They are RNA dependent DNA polymerases, right? They synthesize DNA using RNA as template. So they are called as reverse transcriptase. So there are long terminal repeats, and there is an enzyme for integrase, which is similar in function to that of transposase. And you also have another gene for reverse transcriptase. There is one more, the third category is called as poly-A retrotransposons. It has two open reading frames, ORF1 and ORF2, and there are 5' UTR. UTR stands for untranslated region, and because it's on the 5' side, you call it as 5' UTR, and the downstream one is called as 3' UTR. The difference is that there is a poly-A tail. That means the numerous adenines are present here. Here it's only shown as 3, but it can be up to hundreds. Okay, or 100 maybe. So this is what it has, and this poly-A tail is important for integration. So we'll quickly have a look at this one, mechanisms of retroviral integration. So what happens here? This is the last slide. So you can, here is what we have. The first one is LTR and LTR. Those are the sequences that are recognized. And within one of the LTR, there is a promoter. P here is for promoter. From the promoter, the the transposon is transcribed, so it will form RNA. And because this is forming RNA, you call this as retro. Everywhere you have involvement of or of an RNA intermediate, then you call it as retro. Retro is back. If you remember central dogma, we will study it as DNA, RNA, and protein, as if there is a unidirectional. If you are moving from RNA to DNA, that is moving backwards, or they call kind of representative as something like retro. Okay, so I also told you that there are two genes in it. One of it is the integrase, and the other one is the reverse transcriptase. So those genes are expressed, I mean, in the mRNA, they are translated, and you will have formation of integrase and reverse transcriptase. The reverse transcriptase will work on the mRNA and will synthesize a double-stranded DNA molecule. That DNA molecule is processed, and there is a generation. There are this 3' OH will make a nucleophilic attack on the target site. Okay? This is, well, we'll come to that. So the target site is attacked by the transposon, and then it integrates by the enzyme integrase. Okay, this is the function of integrase. And eventually the gaps are repaired, so you will end up forming the target site duplication. We'll quickly go through it again, once again. So we have the retrotransposon, which has the retrotransposon has gene for integrase and a gene for reverse transcriptase. It has LTR, long terminal repeats on either side. And there will be target site duplication that is not essentially the part of the transposon, but it forms because of transposition. So upon transcription, there is a promoter on this side of the LTR, and the RNA is produced. That RNA can be translated to produce integrase as well as reverse transcriptase enzymes. But the RNA will also be made into cDNA. I think it is complementary DNA, double-stranded complementary DNA by reverse transcriptase, and the reverse transcriptase then the integrase will process the ends of this cDNA, double-stranded cDNA that is produced by reverse transcriptase, and that makes an attack on the target site. Upon that, it gets integrated something like this, and you have these gaps and extensions which are repaired, gap repair, and ligation happens, because of which you have target site duplication. But the important thing is that the genome had only one, if the genome had only one reverse retroviral transposons, now it would have had second one. First one is remaining in the same location. RNA has formed and it moved into a new target site, copied itself or integrated itself into a new site. So there are two transposons in that genome. So it is kind of jumping jump. You can think of it as copy-paste mechanism, right? So these are the mechanisms of transposition. In tomorrow's class, I will describe about poly-A retrotransposons, and that's the end of. There will be a few examples that we will discuss, and then what I want you to prepare for tomorrow is trying to know what are antibodies, how they are formed. This is how they are formed and what they are. I'm not going to discuss about the biological relevance of antibodies. What I'm interested is how do they form. So you must be learning or hearing, at least in light of COVID-19, about antibodies, vaccination, and so on, isn't it? So the first point is to be able to appreciate immunology. You have a molecular or genetic basis for it. How does a cell produce a large diversity of antibodies? So in that, please try to note down what I want. I'm not going to discuss in all these things, but know the structure of an antibody. It has 2 heavy chains and 2 light chains like this. You must have learned those in your plus 2, or even in biology for engineers, you must have read. How is it relevant in this particular course is about trying to know about the recombination, right? A light chain has several parts. It has variable regions, it has joining regions, it has constant regions, and so on. And then how diversity comes about. I will teach this tomorrow. If you understand, it's very good. If you don't understand, it is all right. But I hope you will try to find out some resources on YouTube and try to learn a little bit about it before you come for tomorrow's class, so that we will all benefit from it. Okay? Yes, any questions?